Now for the last session of today's meeting. So we've had our AGM and some excellent talks so far. Now we're on to the Frits Sobels Award. So I'm going to present this, a few slides around the awardees' career to date, and then pass it on for the award lecture. Hopefully, if I can navigate my computer. So the awardee is in very good company, the Frits Sobels Award 2021. And you can see the good company that the awardee will be in line with. So last year, Stefano, we've got other people on the call, such as Andrew Collins, David Phillips as well, Michelin. You can see these excellent scientists, Barbara Dudek, Eugenia Dogliotti, all these guys that we know are top people in the field. And this is the highest prestige level that we have within our society to give to someone who we deem to deserve it. And that person today is Bernd Kaina. So Bernd Kaina, I'm going to present a few slides before you do your talk. So he's an excellent scientist. There was lots of discussion around his career to date being at the top of his field. And I'll just go through some of those achievements in this next slide set. So the 2021 Frits Sobels Award goes to Professor Bernd Kaina of Mainz University. I will go through these quite quickly, mainly just so I can show some photos of Bernd is the plan. So he did his degree a few years ago. He did his PhD in 1976, graduated from the University of Halle, and started doing some publications in this area that we know and love Bernd for. Things like these alkylating agents, DNA damage, DNA repair, carcinogenesis from the onset. So Bernd's been excellent from the onset in this area, and we'll see throughout his career that he's been developed as a career scientist in DNA repair, mutagenesis, and leadership as we get through to later stages of his career. And there's Bernd popped up as well. So hello. Here is his postdoc in Gattersplan and became a working group leader where he developed these leadership abilities and started pumping out ridiculous amounts of publications, as we'll see in a minute. Then in 1984, EU scholarship in the Netherlands, so out of Germany for a bit, and then back to Heidelberg in 1984 to 1985. So moved around Europe, developing his skills in DNA repair and mutagenesis, and really becoming one of the leaders in the whole world in this topic. And then Heisenberg Fellow, the German Research Foundation, and back to Karlsruhe, where he became the head of the working group. And also, I like to do things back to front. This was nominated by the GUM German Mutagenesis Society and supported by many people within that society, including Marcus Christman and Roland Frotzschel and others. So then became the professor at Mainz University, head of Department for Applied Toxicology in 1993. And just before that, in 1990, got his tenureship within the Mainz University. During that time, pumping out lots of amazing publications. What most people, including myself, will recognize Bernd for is his outstanding achievements and being really the go-to person for MGMT and alkylating agents. As you can see, putting out a lot of publications during his time between 93 and 2004. And he's unbelievable. He is the man for MGMT. You can see he's not a one-trick pony. He's got lots of other publications and expertise in these other DNA repair areas as well. But he really is an amazing scientist. I had the privilege to work with him on a couple of publications as well, which was fun too. And then a professor in toxicology, the C4 managing director of the Institute of Toxicology at Mainz University. And here's just an outline of his high achievements at that time, becoming the most highly cited toxicologist in the area, in Germany, Austria, and Switzerland. A huge achievement because the citations in this area were booming at the time, and he was the top of the top. Now for some photos. So here's some of Bernd's colleagues. He's been carrying out these conferences, arranging conferences, and having a big network of leading experts in DNA repair for many years. People such as Leonis Sampson on his right arm there, Jeff Margerson, Grigory Dynov, and Gerard Fritz, who I think did a PhD with him as well before, as well as many other top-end faces that you'll see, not just for that conference, but many people who he worked with on a regular basis too. Here's another one. the German Symposium for DNA Repair and Human Disease, people like Andrea Hartwig, Wilhelm Bohr, Alexandra Burkle, and just really just showing the network of Bernd and his ability in this area at the scientific level, but also at the high accolade of people recognizing him and going to his conferences on this topic too. So he also achieved an award, the GUM Award in Potsdam 2010. This is Hans-Jörg Martus presenting it to him, doing a nice presentation there. When I went to visit Bernd, I saw that he had lots of nice DNA damage paintings that were around his laboratory. I thought this was very nice. These were commissioned by his friends, by local artists. And this was a very nice touch that I saw in his top-end laboratory as well. And then went round the Chagall paintings and stained glass around his local church. And this very nice conference where we went and sampled some very nice wine and sparkling wine. I did a presentation. There's Roland there as well, and some people at the mines. I think Roland's there. We got Helga's there in the bottom right too. So I just like showing photos in these sorts of award presentations too. I think for the high accolades of Bernd's research career, it's good to go towards the publications from the start and see that Bernd's published over 300 publications, huge amount of publications, over 18,000 citations. This is huge. This is the top of the top in any field. H index of 75, meaning he's got 75 papers where 75 or more people have cited him as well. And an H index is a good way to compare different academic scientists throughout the globe. And really, people in the DNA repair field and people in mutagenesis and carcinogenesis realize that Bernd is such a leading expert in this area. And we are very fortunate to have him within our society. And it's an honor to be able to present him with this award today. So very happy to see you here, Bernd. I've loved working with you today. Recently, for people like the EMA as well, we do bits and bobs. And for the Threshold Working Group, the German Threshold Working Group was fun too. But yeah, it's my honor to provide you with the EMGS Frits Sobels Award 2021. And yeah, over to you, Bernd. Thank you very much for being such a nice guy, top scientist, and over to you. Thank you very much.
Okay, thank you, George, for your very kind words, for your very kind introduction. And I'm really thrilled about these very nice pictures, where you have them from. So it was really very, very interesting for me. Actually, I started working with alkylating agents quite a long time ago, 1972. And you mentioned the first paper I wrote myself. This was actually the part of the thesis published in 1975. And after almost 50 years in this alkylation business, a lot has come together. So I'm very pleased to receive this high award from the EEMGS, especially also since I was able to get to know Professor Sobels personally. I met Professor Sobels for the first time 1984, after I immigrated from East Germany to the West. And my goal was immediately after arrival in West Germany to move on to Leiden. Because there was a department of radiation biology and mutation research, which was at the time leading in the field of molecular dosimetry of alkylating agents. Sobels was a director of this institute, and I remember the first conversation with Sobels in his office. And I have to say that what Brian Bridges wrote in his obituary for Sobels, that this is actually true. All who worked with him and came to know him will count it an honor. So this applies to me as well. During my first visit at this institute, Professor Sobels invited me to report of my work, and I gave a seminar there. And I still remember the seminar room in this famous institute with these very famous guys and ladies as well, including Margaret Chinichka sitting there. And I decided to present the data I obtained before as a young postdoc at this institute in Gartersleben, where we performed experiments with Chinese hamster cells. And we treated Chinese hamster cells with methylnitrosourea This was basically the continuation of my thesis with methylnitrosourea, 1 cell cycle, 2 cell cycles, or 3 cell cycles before fixation. And what I observed was that in this cohort, if cells were treated 1 cell cycle before fixation, sister chromatide exchanges are not formed at all. One needs 2 or 3 cell cycles in order sister chromatide exchanges to be formed. With very high concentrations, one can also achieve the induction of SCAs in the first cell cycle. So this is completely different if you use, for instance, mitomycin C or UV light, which are able to induce sister chromatide exchanges in the first cell cycle at the same frequency as in the second cell cycle. So there's something specific on methylating agents that allow them not to induce SCAs in the first cell cycle. So at this time, it was well known that these alkylating agents, as so-called SN1 and SN2 alkylating agents, target DNA at 12 sites. These are today, there's 14 sites. And it was also known that most of these sites are repaired by base excision repair, by MGMT, and by B homologous proteins, which we are not yet discovered at this time. MGMT repairs 2 sites, O4-methylthymine and O6-methylguanine. This is a very, very minor site. So usually all of the work, people focus on O6-methylguanine, which amounts to about 6-8% of total alkylations for SN1 alkylating agents. So at this time, interestingly, it happens just that during the same week when I was visiting this institute, Professor Mitra from Galveston or Oak Ridge at this time was visiting the institute as well. And Professor Mitra was leading at this time. He has established several assays that allowed to determine the enzyme activity of MGMT, of this repair protein. And I kept in touch with Shankar Mitra, and in Leiden, I started immediately then a project on cloning of MGMT. Several other groups did it as well, including Peter Karan and Sekiguchi in Japan, but he made the race. So Shankar Mitra was the first one who succeeded in cloning by Kesutano. So I did it as well, but he got the sequence, he published it immediately, but he also shared the sequence with me. This was the first publication of the MGMT cDNA. So I got the sequence from Shankar Mitra, and we started a wonderful collaboration over years. So I prepared a cDNA expression vector, which is still in use now and interested by several groups using it for generating transgenic cell lines and also individuals. So having transfected Chinese hamster cells, we can see that the sister chromatide exchanges in these cell lines that express MGMT at different amounts declines to nearly 0 level, dependent on the expression level of MGMT. The mutation frequency declines to near 0, and the cell survival increases to nearly 100%. And this was the first clear indication and evidence that O6-methylguanine is responsible for SCAs, for point mutations, and also for cell death, for cell killing by these alkylating agents. So 6-methylguanine is a genotoxic and killing agent, and we concluded MGMT is a keynote in protection. By the way, this is clearly dependent on the amount of MGMT molecules per cell, and this data implicates at the same time that the threshold for genotoxic effects or for mutations, for instance, or for SCAs, and also for survival, is determined by the amount of MGMT present per cell. We extended this work by expressing the human MGMT gene in keratinocytes of mice. So these are wild-type mice, these are transgenic mice, and if we treat these mice with a single dose of methylnitrosourea followed by repeated TPA treatments, then we can see the induction of papillomas of tumors in the wild type and much less in the transgenic mice. We can also convert these papillomas into carcinomas. This is a process of progression, and we can also see that the progress of progression is significantly reduced in transgenic mice. So indicating O6-methylguanine is a tumor initiating and a tumor progression lesion. It is not a tumor promoting DNA damage, because tumor promotion was unaffected by MGMT expression in transgenic mice. Mice express MGMT, but at low amounts, so we overexpress the MGMT. On the other way around, we can knock down MGMT. The colon express quite high amounts of MGMT in mice, but human more, but still expressing in mice. So in this case, we knocked MGMT, we knocked it down, and by means of mini coloscopy, we were able to inspect the colon of mice. This was done in collaboration with Leona Samson, from whom we got the mice. But this coloscopy method was established at the Institute of Toxicology in Mainz. And here we saw we were able to measure the amount of O6, the amount of lesions, carcinomas, and here's a small carcinoma lesion. And if we quantify this, we can see that for MGMT wild type cells, wild type mice, there is no tumor formation in the colon. At low dose, in the low dose range, one needs higher doses in order to induce tumors. But in MGMT knockout mice, this is a blue line, there's a clear linear increase in tumor formation. With this high dose, the mice did not survive anymore. So this indicated again that O6-methylguanine is also a powerful carcinogenic DNA damage work that has been done in collaboration with Jörg Fahrer. He was at this time working as a group leader in my institute in Mainz. So this indicated O6-methylguanine is a significant mutagenic, genotoxic, and carcinogenic DNA damage. It was very interesting to see that O6-methylguanine has also such an impact on survival of cells. I still remember the first discussion in 1984 in Leiden, when Bernd van Zeelen asked me, is O6-methylguanine also a cytotoxic DNA damage? And my answer was immediately not. It is no. Because if it's toxic, it cannot be at the same time mutagenic. But this is human thinking, so it was absolutely not the case. Today we know O6-methylguanine is also cytotoxic DNA damage, and this led us to turn with most of our activities to anti-cancer drugs. Because these alkylating SN1 anti-cancer drugs are being used for treatment of especially brain cancer patients. And here in tumor therapy, we need toxicity, cytotoxicity, not that much genotoxicity and mutations. These are unwished side effects, but we need cytotoxic effects. So therefore, it was quite important for us to elucidate the pathways that lead to cytotoxicity triggered by this specific DNA damage, O6-methylguanine. Here we can see that the apoptosis is reduced according to the MGMT activity in malignant melanoma cells. And we were also the first to show this for melanoma cells and also for glioblastoma cells, that O6-methylguanine is inducing apoptosis. These are not transfected cells, these are different cell lines expressing MGMT to different extent. What is a mechanism of O6-methylguanine triggered toxicity? A breakthrough paper was actually this one published by Paul Mortridge's group. And he published and reported that cell lines chronically treated with MNNG became resistant to MNNG, and he designed this or named this an alkylation-tolerant phenotype or alkylation-tolerant cells. And in his summary, he writes, these findings substantiate the idea that specific mismatch repair contributes to alkylation-induced toxicity and implies that this process serves as a barrier to mutations. I think this was a very wise notion to not only to highlight the finding that O6-methyl-guanine is cytotoxic, but that this process may also serve as a barrier to mutations by eliminating cells that harboring this particular DNA damage. Still, it was not known how the cells actually undergo cell death, could be necrosis or something else, and therefore we looked carefully at the apoptotic response, and this was the first paper reporting that O6-methyl-guanine triggers apoptosis. Leonard Sampson and Boston published at the same time absolutely the same data, so it was clear from this time on O6-methyl-guanine is a powerful apoptotic DNA damage. In this paper, we published and or we reported about the pathway leading to apoptosis. So the mitochondrial pathway was activated, but most important is we measured also at this time DNA double-strand breaks. By the Newton Comet assay, gamma H2X was not yet available at this time, but found that clear double-strand breaks were induced and occurred preceded in the wave the onset of apoptosis. So double-strand breaks are an intermediate in this particular pathway, and this led us to postulate that O6-methyl-guanine is a primary lesion, secondary lesions are formed, and double-strand breaks are intermediate lesions. We defined it as tertiary lesions. And in Chinese stem cells, this particular pathway becomes activated, but this is not generally true. It depends then on the cellular background. This was basically substantiated by Steve Quiros, a PhD student from Costa Rica in my lab, and he synchronized China stem cells and created the synchronized cells after by my mitotic shake off by my MNNG treatment immediately after seeding. And we can see that the synchronized cells goes here to the first cell cycle. This is the second S phase, and they will look then at the gamma H2X foci as an indicator for double-strand breaks. We can see that 1 day after treatment, this is this time period, double-strand breaks are formed. So double-strand breaks actually need 2 cell cycles to be induced at high levels. In MGMT transfected cells, nothing happens. So this clearly shows that these double-strand break induction with this type of treatment under these conditions, and the dose is always very important in these experiments, is due to O6-methyl-guanine triggered by this particular DNA damage. If we measure then the cell death by apoptosis, we can see that cell death occurs later, starting at this particular time. So we have double-strand break formation here, and it obviously the pathways then leading to cell death need some time to be fully activated in order to see then the formation of or the appearance of apoptotic cells. So this is a model that we proposed and published and was, I think, generally accepted. There was another model indicating direct activation of pathways, but this model was not substantiated by any work. So this model is still holds true. We have here O6-methyl-guanine, and this is a sequence leading to mutations and cancer. And this is a sequence leading to toxicity and genotoxic endpoints. And the model states that O6-methyl-guanine mispairs with time in the first round of application. And this mispair is recognized by mismatch repair proteins, mutase alpha and mutase beta. So these 4 proteins bind there. And this gives rise by the mediation of endonucleases that that gaps are formed. So single strand breaks are induced here, and this results in long stretches of gaps. Originally reported about 1,000 nucleotide long gaps. And of course, these long gaps block replication in the second replication cycle, and then things happened. And we showed, and also first we postulated, that this particular structure can be tolerated by cells by a process called tolerance of O6-methionine via recombination bypass. So recombination takes place at this place, and including several players, and this leads to sister chromatide exchanges. So according to this model, SCAs are not toxic events. They are outcomes of a process that helps cells to survive and tolerating O6-methionine secondary structures. On the other hand, double-strand breaks can be formed at this time, and this structure, and these double-strand breaks finally trigger apoptosis, also aberrations. So what are the players then involved in causing drug resistance or protecting against this whole process? And one of these is MGMT. MGMT, of course, is a main key defense mechanism because it is on the top of the whole pathway. MGMT removes very quickly in the suicide reaction O6-methionine from DNA in an error-free fashion. And as I said, the downstream effects depend on the amount of MGMT per cell, because this is a suicide reaction on the expense of the enzyme, of the suicide enzyme. So this prompted us then to look at the expression level of MGMT in normal tissues and in tumors. And impressively, MGMT is extremely strongly regulated. It is very low expressed in glioblastoma and brain cancer, and high expressed in other tumors, including lung and ovarian cancer. And interestingly, for this group of tumors, these SN1 methylating agents have been applied. Before everything was known, metronidazole was already applied in the 50s in Russia, for the Soviet Union, for treatment of glioblastoma before temozolomide and other methylating drugs were available on the market. So this is a reason, the low expression level of MGMT, why glioblastoma respond more or less to these alkylating agents. The other finding was that MGMT is extremely variably expressed. We have here breast cancers and individual tumors, and we can see there are tumors expressing MGMT at a very high level, and there's a subgroup of tumors not expressing that MGMT at all. And the same is for brain tumors, but the variability is much, much lower. On average, expression level is low, and also there is a group of tumors not expressing MGMT. In the primary tumors, we did with a large cohort of patients, we found that about 17% of glioblastomas do not show MGMT expression at all. MGMT is clearly correlated with survival response of the patient, or with a therapeutic response of the patients. Here we measured MGMT, we measured the therapeutic response, so overall survival of patients, and divided these groups into cohorts with low and with high MGMT in the tumor. So we were able to define a threshold of about 30 femtomole. So this is in this range where the difference is significant. So the MGMT activity determines the survival outcome of glioblastoma patients, although the overall prognosis is very, very low and bad for these patients still. MGMT is strongly regulated, and this occurs by promoter methylation. So therefore, it was important to correlate MGMT activity to promoter methylation, and actually there is a very strong correlation. The MGMT methylated, CpG methylated promoter, these tumors show very low MGMT activity compared to the tumors harboring the unmethylated promoter. And MGMT promoter methylation is now worldwide being used as a method of stratifying glioblastomas. This shows the same Kaplan-Meier curve here, survival response of the patients having the methylated promoter, so the MGMT is more or less silenced, and the unmethylated promoter. And these studies were done by many other groups. First one by Monica Hegi, just now one of the most often cited paper. So we found that 70% of gliomas do not show MGMT activity, 40% displayed MGMT promoter methylation, and MGMT activity and promoter methylation are highly predictive in high-grade gliomas. So MGMT promoter methylation is a predictive marker in personalized brain cancer therapy, and this is now worldwide accepted. If patients are treated with temozolomide, this is a methylating agent which acts on the DNA level. It's a triazine, not a nitrosourea, but acts exactly like methylnitrosourea and other SN1 alkylating agents. So therefore, we could translate everything what was known at this time about the regulation also to temozolomide. It was well known at this time due to the background work of Jacques and Françoise Laval, to Margeson in Manchester, and to Anthony Peck in USA, that if you treat rats with any type of genotoxic stress, even radiation or other carcinogens, that the enzyme activity increases in the liver. But it was not clear, is this enzyme activity, increase of enzyme activity just due to stabilization of the protein? And therefore we were interested in figuring out, is this due to gene activation? And with the availability of MGMT, cDNA, we were able to do northern blots. So at this time, everything was, today we use a lot of Western, but gene expression studies were done by northern blots. So this was a classical method, very time consuming and also a little bit dangerous because you need a radical probe. And using this method, we were able to show that MGMT gets upregulated if cells were treated with genotoxic agents in rat hepatoma cells. So everything has been done in rat hepatoma cells. And by the way, we also measured mutation frequencies in H4 hepatoma cells in cells not treated or pre-treated with a low dose of MNRG or X-rays, but then challenged with a high dose. And if these cells were pre-treated, the mutation frequency was lower than compared to the MNRG challenged fraction, indicating that pre-treatment, which induces MGMT, has a significant effect on the mutation frequency induced by the challenge dose. And this was the first indication and evidence that indeed mutagenic adaptation or something like a mutagenic response can occur in mammalian cells. But in this case, it happens only in rat liver cells. We did similar experiments with human fibroblasts or with HeLa cells. We couldn't confirm it, so we couldn't find any upregulation of MGMT also in human fibroblasts. But this still left the question open: can MGMT be induced as a subject of induction in glioblastoma cells, in tumors? And this is an outcome of a long story, a long journey in this field. In our hands, we couldn't find any evidence that temozolomide and radiation is able to upregulate MGMT in glioblastomas. But what we found was glucocorticoids have the ability to upregulate MGMT, and actually the promoter of MGMT contains glucocorticoid responsive elements. So therefore, the promoter can be activated by dexamethasone or other glucocorticoids. It contains a lot of SP1 sites, and this is presumably the reason, compared to the rat promoter, why the human MGMT promoter is not induced and not upregulated by genotoxic stress. But interestingly, glucocorticoids upregulate MGMT, and nevertheless, they are routinely used in GBM therapy. So this indicates that this is counterproductive, and there are indeed studies showing that glucocorticoids can reduce the therapeutic response of the patient. So it may be a counterproductive treatment schedule. What can we do if MGMT is expressed at high level, which also occurs in glioblastomas, especially in recurrences? There are several strategies, and one is to use MGMT inhibitors. One is 0,6-benzoguanine, which is a pseudo-substrate of MGMT. It diffuses into cells, or will be taken up actively, and inactivates MGMT immediately. So this inhibitor was used on many experiments in vitro and also in vivo, and also in clinical trials. But unfortunately, there was no benefit up till now, presumably because the systemic toxicity of the alkylating anti-cancer drug is then too high. What we need is a targeting of this DNA repair inhibitor, and this is one approach in targeting 0,6-benzoguanine. This was a patient, a tumor patient, suffering from a glioblastoma, and we can see that the MGMT activity went up in the recurrence and the second relapse as well. And therefore, we decided to treat them with this particular approach by injecting directly 0,6-benzoguanine. So this was a local regional treatment approach, but this is not yet established in the clinic. Other way is to inhibit these inhibitors to some vehicles that are taken up selectively by tumors. We thought a reasonable strategy could be to bind 0,6-benzoguanine or 0,6-bromotenylguanine to glucose. With the hope behind that tumors express a higher level of glucose transporters. And what we can see here is that this was reported in these papers. These are very effective in inhibiting MGMT. They are very effectively taken up in cells, but there was no selective uptake in glioblastomas because the uptake occurs by flipases and type 4 ATPases. So this is a complex uptake mechanism. However, it might be that other tumor groups are responding, and simply clinical trials are needed using this type of targeting approach. So this were a few words on MGMT. What about mismatch repair? Mismatch repair it is important here to note that quantitative changes of mismatch repair do already have an impact on the response of cells. We have here isolated several cell lines expressing MSH2 at different levels by chronic treatment. And if we measure the response of these cells, we can see that the MSH2 expression is inversely related to the resistance level. So high MGMT expression high MSH2 expression causes a high level of sensitivity. So this quantitative correlation indicates that also mismatch repair might be a reasonable player in generating toxicity or drug resistance in patients. And actually, there's a paper by Lerner Samson, and there are many other papers showing that mismatch repair is regulated in gliomas, and they express different levels of mismatch repair. And they report that even minor changes in the expression of mismatch repair have an impact on the glioblastoma response to temozolomide. But this even needs to be substantiated with other clinical studies. It is quite interesting that if we treat cells with a single dose of temozolomide, that we observed a decline, a decrease in the level of mismatch repair proteins. On RNA level is MSH2, MSH6, also XO1, which may indicate that during chronic treatment, mismatch repair becomes, due to the treatment, downregulated. And this may cause then resistance of cancer cells, and because they are simply protected, we need mismatch repair in order for eliciting these cytotoxic responses. Now, let me say a few words to double-strand break repair. Double-strand break repair is downstream, and we have seen double-strand breaks are formed, so-called tertiary lesions, but there are different mechanisms that might be involved: homologous recombination, anomalous end joining. Which one of these pathways is really involved and important? So we use different Chinese hamster mutant lines and found that for non-anomalous end joining, there is no or only very small difference in the apoptotic response following treatment with temozolomide. But if we use homologous recombination mutants, there's a huge difference in the response. So the mutants are significantly and much more sensitive than the DNA, than the non-homologous end joining mutants, indicating that homologous recombination is required for repair of double-strand breaks, and this is the main pathway leading to drug resistance or to protection against the toxic effects of generally of these agents. We extended this work and translated this to glioblastoma cells by downregulation of RAD51 and established different stably expressing cell lines. And again, we found here a quantitative correlation between the RAD51 expression level and the amount, the dose that kills cells, meaning the level of resistance of cells. The higher the homologous recombination level is, the more the cells are resistant, indicating again that it is worth to look at quantitative expression levels of homologous recombination proteins in tumors. It is well known that homologous recombination impairing can be harnessed for something like synthetic lethality by co-treatment with PARP inhibitors. And this happens here as well. So we are therefore interested in downregulating homologous recombination by RAD51 and at the same time by inhibiting PARP. And if we use this type of approach, then we can see that the toxicity can be strongly ameliorated in this particular case, indicating synthetic lethality under the prerequisites of downregulation of homologous recombination is a reasonable approach also for cancer therapy of gliomas. Okay, we measure then the expression or the impact of other players of homologous recombination, including XRCC3, which might be here, which we propose to be a novel target in glioma therapy. And overall, to make a long story short, there are many players involved in homologous recombination, including XRCC3, BRCA2, RAD51, Werner's XRCC2, and maybe all that are involved in homologous recombination or even more contribute to the outcome, contribute to a given level of drug sensitivity or drug resistance. DNA repair by homologous recombination competes with a DNA damage response. And it is well known that this structure, this secondary structure, can give rise to activation of ATR check1 or ATM check2. And so we are addressing then the question is, how important is this pathway in survival or in death responses? So is this actually a survival or death response? We did it by downregulating ATR and ATM or by inhibiting the corresponding check proteins. And we can see that if we downregulate ATR, the cells become much more sensitive to temozolomide. If we inhibit check1, which is activated by ATR, cells also become more sensitive. This happens also for ATM and check2, but not to the same extent. In MGMT proficient cells, nothing happens. So this is actually part of the O6-methyl-guanine response. And this led us to conclude that the DNA damage response triggered by ATR is actually a survival pathway, presumably by activating enhancing the level of homologous recombination repair. On the other hand, we know that ATR and ATM target p53, and p53 becomes phosphorylated at the same time and becomes active. What is the role of p53 in this game? So p53 is a little more complex. We have here ATR and ATM activated in this case by O6-methyl-guanine secondary lesions. And this activates check kinases, and finally p53 becomes phosphorylated, stabilized, becomes transcriptionally active. But p53 can be phosphorylated at different sites, including serine 15. And there is another pathway, and this one targets SIA1. SIA1 becomes phosphorylated. This is an inhibitor of this kinase HIPK2. And if SIA1 becomes phosphorylated, it becomes degraded, HIPK2 becomes liberated and released. HIPK2, active HIPK2 phosphorylates p53 at another site. So we have here serine 46. And we have here serine 15 and other serines phosphorylated by different pathways. And this is supposed to be a cell death pathway. So is this also the case? Is this true for glioblastomas? And this was done by Yang He, a PhD student in my lab. And if we downregulate HIPK2 by an antisense strategy, the level of apoptosis is clearly reduced, significantly reduced. If we, on the other hand, downregulate SIA1, the level of apoptosis is enhanced, strongly indicating that this model holds true. HIPK2 is required for inducing apoptosis, maybe not completely, but it ameliorates at least the amount of the apoptotic yield. And SIA2, the inhibitor of HIPK2, for the opposite. So this model can be applied also for gliomas, and for the O6-methyl-guanine response, SIA1 and HIPK2 triggers the phosphorylation of p53, and p53 in turn then transcriptionally activates several genes, including the FAS gene. And actually we can see if we downregulate HIPK2, the death receptor FAS is downregulated, and also p10. And these are pro-apoptotic genes and players. So this is a new pathway which is playing a role in gliomas. p53, however, is a little bit more complex. Because p53 does not play a role only for methylating agents, but it is also involved in the response of O6 chloratolytic agents. O6 methylating agents like temozolomide are being used first-line glioma therapy, and these O6 chloratolytic agents are second-line chemotherapeutics. One of these is BCNU, classical O6 alkylating agent, also used for many other tumors, and CCNU lomustine. What are they doing? They also alkylate guanine at the O6 position, forming not O6-methyl-guanine, but O6-chloratoguanine. This damage, this adduct is also repaired by MGMT, removing this chloratoguanine. But O6-chloratoguanine is not a stable lesion. It undergoes a rearrangement intramolecularly and leads to N1 guanine and 3-cytosine crosslinks. And these crosslinks are doing the job. They are highly toxic. So if we compare now the toxic response, the apoptosis level induced by temozolomide for p53 wild type and mutant, we can see that wild type is more sensitive if we treat with temozolomide, but is more resistant if we treat with ACNU, with these chloratolytic agents. What is the reason? Because this apparently dichotomic, completely opposite effect of p53. So the reason is simply for methylating agents, p53 upregulates the death receptor FAS, which is shown here in the same cell lines that were used for survival experiments, whereas for the chlorinating agents, p53 upregulates other DNA repair genes such as DDP2, which we can see here, or XPC, and also the polymerase, DNA polymerase eta. And these are DNA repair proteins that obviously are upregulated and are under control of p53, which protect cells if the cells are treated with O6 chlorinating agents or any other adducts, including also benzpyrene. Also for these big adducts, this pathway plays a significant role. So we have here the ATM, ATR, kinases that activate p53, and in turn p53 upregulates DDP2 and other repair enzymes, not in human cells significantly MGMT. And this is a tolerance or a repair pathway, whereas if this pathway is activated, the FAS receptor, then the cells die. And this is most important for the methylating SN1 methylating agents. And this is important for the O6 chlorinating agents. And therefore, we see these different responses, which has a strong impact for cancer therapy, because now it would be indicating that it is worth to stratify patients according to the p53 status. So, O6-MET2 this is what we learned from all of this work, is a highly toxic DNA damage leading to apoptosis. But we know that also other endpoints can be triggered by genotoxic events, such as autophagy and senescence. What about this? We measured, of course, also autophagy in glioblastoma cells after treatment with temozolomide, and we can see autophagy is induced with this low dose range. These are therapeutically relevant doses at high level. And we can also see that senescence, cellular senescence, is induced at high level and with the same low doses. So this indicates that O6-MET2 is a potent trigger of autophagy and senescence. The control is in MGMT transfected glioma cells, nothing happens. So these effects are brought about, these are isogenic cells, these are clearly brought about by this particular DNA damage, O6-MET2. And we also learned that these endpoints are interrelated. There's a crosstalk between autophagy, apoptosis, and senescence. If we inhibit autophagy, then we can enhance the level of apoptosis, indicating that autophagy is is a protective endpoint, is a protective pathway. It blocks autophagy, at least when these low concentrations, therapeutically relevant concentrations of temozolomide are being used for treatment. Interestingly, if we look at the dose response for these endpoints, we can see that apoptosis, senescence, and autophagy increases linearly with dose. Senescence, the outcome of senescence with the same dose is much higher than apoptosis, about double of what we observed if we measure the outcome of apoptosis, and also autophagy increases linearly. This indicates for these endpoints there's no threshold in MGMT not expressing cells. This is always important to mention. There's no threshold in these responses. And the second point, very important to note, is apoptosis is not a major pathway. Senescence is a major trait and a major outcome after treatment with these SN1 methylating anti-cancer drugs. We continue this type of studies. This is being in press now by measuring the amount of O6-MET2 in the cells. And we can see that the amount of O6-MET2 is linearly increasing with temozolomide dose. This is a therapeutic relevant dose range, not in MGMT expressing cells. There's a slight induction, obviously, due to saturation of the repair system. And now we were able to correlate the amount of O6-methylguanine directly to a particular endpoint, such as DNA double-strand breaks as measured by gamma H2X or 53BP1, which increase linearly with a dose of temozolomide. And this shows the correlation between the DNA double-strand breaks, apoptosis, and senescence as a function of O6-methylguanines per cell. And the outcome is quite surprising. For 20 micromole temozolomide, which is a serum concentration, achievable serum concentration in patients after treatment with temozolomide, about 20,000 adults are induced, which corresponds to 66 gamma H2X foci, 20% apoptosis, and major outcome is senescence. Or in other words, if we recalculate, about 400 O6-methylguanine adults give rise to 1 double-strand breaks. 400 adults, 1 double-strand breaks, and many years ago, we published that 30 O6-methylguanine adults give rise to 1 sister chromatidic changes. So the induction of sister chromatidic changes occurs at much higher frequency. The probability is higher to be converted here by O6-methylguanine. We need 10-fold higher adult levels in order to be able to induce a single double-strand break. So let me summarize the data at this point. This is a main pathway leading to point mutations, assays, aberrations, and cancer. This is a main pathway leading to apoptosis if the mitochondrial pathway gets activated. We also showed that the Jun kinase pathway can be activated, and this leads to upregulation of the Fas ligand, which supports this pathway. Here we have upregulation of Fas receptor by P50 Fas ligand, and Fas receptor strengthens and the induction of this particular pathway. But we also learned that the DNA damage response activates the HIPK2 P50C046 pathway, leading to or supporting the induction of apoptosis. On the other hand, we know by now that sustained activation of this response, which occurs actually in cells, in glioma cells that were treated just with a single dose of temozolomide, activates the ATR-ATM pathway by activation of cyclines and P50C P21 pathway, and this leads to G2 arrest. And if this happens over long periods of time, so if the DNA damage response is permanently activated, this leads to the induction of senescence. And actually after, let's say, 8 or 10 days after treatment with temozolomide, with a single dose inducing O6-methylguanine, after 6 days, we achieve here the induction of apoptosis, and after 10 days, a huge amount of cells that undergo senescence. And these cells exhibit also the senescence-associated secretory phenotype. This means they secrete pro-inflammatory cytokines, and this may be a sequence, a pathway leading finally or enhancing the formation of cancer or supporting the progression of tumors if this happens during tumor therapy in cancer cells. So we have here the senescence pathway. This is the main pathway leading or the main outcome of O6-methylguanine activated pathways. And therefore, we are interested now to eliminate senescent cells. So how can we do this? There are agents, synthetic or small molecules that eliminate specifically senescent cells. And this is being done currently by Leona Belzig, I guess, last PhD student in my lab. And she showed, now this is not yet published, that senescent cells can be selectively killed compared to proliferating cells. And this may also happen if natural compounds like fisetin are being applied. Fisetin appears also to be such a selective xenolytic agent. So if we eliminate senescent cells, the hope is that we can drive these senescent cells into the apoptotic pathway in order to eliminate them from the population, because there is a suspicion that senescent cells may be reactivated in the tumor, and this may give rise to secondary tumor formation to recurrences at much later times. So finally, let me say a few words as an outlook. I focused on alkylating therapeutics. However, the data can be directly extrapolated to O6 alkylating environmental carcinogens, and I would be really very excited to see if these cause exactly the same responses as temozolomide. I want to pick up just a few questions here, and I believe they are worth to be explored. One is O6-methyl thresholds. Are there thresholds for mutations, cell death, and senescence in vivo for different tissues? And what is the role of MGMT mismatch repair, homologous recombination, and the DNA damage response in vivo in different tissues, triggered by O6-methyl and triggered by O6-methyl induced by nitrosamines and other O6 alkylating carcinogens. So we can directly translate these data obtained with temozolomide to environmental carcinogens. So the question is, are nitrosamines, for instance, also able to induce senescence in vivo? And is there tissue type dependence? And are there individual differences depending on the DNA repair status, for instance, BRCA2 homo or heterozygous individuals? And finally, are there cyanolytic drugs that can be targeted and they directed against carcinogen-induced senescence? We know that benzapirin is very effective in inducing senescence, similar to what I showed for temozolomide. So the DNA adults induced by benzapirin have the same or activate presumably the same pathways as O6-methyl alkylating agents anyway, and also acetoxy-acetylamidofuran, maybe many other carcinogens may be able to activate this pathway. And it would be very exciting to see whether there are thresholds and whether this is a protective or a sensitizing pathway protecting against cancer or supporting cancer formation. All of this is not yet really well known. Sometimes if I look back, it seems to me that the translation of the data to the clinic goes very low. And this is a selfie from the State Gallery in St. Petersburg, the Volga slippers. We need more clinical studies and then more intensive collaboration with clinicians who are willing to translate these data. And of course, the environmental issues are now extremely important to translate this one also to nitrosamines, for instance, tobacco smoke, carcinogens that induce O6-alkyl-guanine, and also foodborne carcinogens that have the ability to induce this type of DNA damage. I would like to thank all colleagues who have accompanied me over the years, postdocs, now they have their own working groups, and also PhD students. My first students, Gerhard Frits, now professor in Düsseldorf, Markus Christmann, professor in Mainz, Mario Timic, also professor there, and Steffen, and many other PhD students were involved. And thanks also to my collaborators over the many years in Mainz, but also abroad, and especially Shankar Mitra in Oak Ridge and Galveston. Thank you very much, and this shows the staff, the Institute 2-3 years ago. All the best to you, and thank you very much for listening. It was a long journey, but we are not yet really there where we should be and where we are. Thank you very much. Thank you, George.
Thank you, Bernd. Congratulations on the award again, Bernd. And that was a lovely presentation showing your career and your amazing publications to date and really getting into a lot of detail about how you are the leading person on MGMT and many other aspects of DNA repair. So thank you for your excellence in this field, and I continue to follow your career and your high impact publications and see how they have huge importance for many stories, such as with the alkyl nitrosamines and everything. So thank you so much for your ongoing work, and again, congratulations. I wonder if you've got your medal with you, could you potentially show your award?
I'm pleased to show you this. We have it here. This is a medal.
Oh, that's lovely.
Award.
Oh, fantastic.
Yeah.
Oh, if we could grab a picture of that for the LinkedIn as well, that'd be fantastic. Thank you so much, Bernd. You're excellent guy to work with, and that was very interesting and very exciting.
I'm very pleased for this one. I must say, if I was talking to Frits Sobels 1984, I still remember exactly this particular chat, because I was asking him for a position. I got another one, and if I would say him, okay, Frits, I will be getting your award named according to you, I think he would be extremely thrilled. Yeah, me too. And thank you.
Oh, fantastic! No, congratulations, very well deserved, and yeah. Excellent that you knew Frits as well. That's a very nice story to finish up on. So I think we'll move on to wrapping up the whole session now. But thank you again, Bernd. That was fantastic, and I love seeing you present, and I love working with you. So that was lovely.